Hi guys, welcome back to my channel. Today I want to break down a passage for you from AAMC full-length 1, the biochem section, passage 1, all right? So this passage itself is not very difficult or challenging. The 6 questions are all fair. As you know, I prefer digging into those really convoluted, challenging passages and questions for you on my channel for you to get the perfect MCAT score. But I feel like if you are on the early stage of studying MCAT, those challenging passages might be a little bit far stretching, whereas this one is really representative of the MCAT logic. And then also this passage represents the graphs, the tables in the way that you will always see on MCAT. So I feel like it's really good material if you're, like I said, on the early stage of studying MCAT. And then we're gonna read this passage and then do all 6 questions, okay? All right, so about 15% of colorectal cancers, CRCS, or colon cancers, okay, so this is about colon cancers, are caused by defects in the mismatch of repair system, MMR, okay? So I am highlighting the acronyms here because for the MCAT passage, they usually prefer using the acronyms, so they would just say the full name for one time, and then later in the passage you would just see MMR, CRC instead of the full name. All right, okay, so the mismatch repair system, MMR, that corrects certain DNA replication errors. Okay, so first sentence is very straightforward, telling us the defects in this DNA replication error correction system, MMR, can cause colon cancers, right? Okay, in some cases, this MMR defect contributes to, okay, this MMR can also cause what? Microsatellite instability, MSI, okay? I don't know what is MSI, but I'll just keep reading, okay? So it says, which is characterized by genetic hypermutability in DNA microsatellites, small repeated sequences that occur throughout the genome, okay? So this is saying that, okay, this MSI is basically some DNA mutation, right? And then they're not just random mutation, they're where it's the sequence where you have those small repeated sequences that mutation can occur, okay? That's MSI. All right, second paragraph. Researchers studying MSI in CRC cell lines and primary tumors observed that 100% of those analyzed contain what? Contain deletion of 3 to 8 base pairs in the 17-thymine nucleotide satellite located in intron 8 of the gene coding heat shock protein 110, HSP 110. Okay, wow, this is a long sentence, right? It has a lot of information, but it's okay. We can just break it down, right? So basically, the first sentence of the second paragraph is saying that researchers, they study some cancer cells, right? Cancer cells. And then they observed that 100% of what they studied tells us that there's a deletion, okay? So this is my DNA. There's deletion of 3 to 8 base pairs, okay? So 1, 2, 3, 4, maybe 4 base pairs. Okay, these can be deleted, okay? And then they are in the 17-thymine nucleotide microsatellite, okay? And then we know microsatellite from the first paragraph, they're those small repeated sequences, okay? And then in this case, it's the thymine that's being repeated. So those 3 to 8 base pairs can be maybe three thymines or 4 or 5 or 8, right? I don't know, 3 to 8, okay? So these can be deleted. And these, where are these located? These are located in intron 8, okay? So intron 8 is the region of this DNA sequence, okay? All right, so this gene, okay, and what does this gene do? This gene encodes the heat shocking protein, HSP 110, okay? All right, so not too bad, right? This strand of DNA, it's our gene, right? It first go through the central dogma, first transcription and then translation. Translation gives us this protein 110, okay? And then those introns are part of the mRNA processing, where before we jump into translation, the introns can be can be deleted, right? Can be cut by the enzymes, and then so the exons can exit the nucleus and have further translation and eventually the protein. Okay, so that's what this is about. Okay, let's keep reading. HSP110 can bind structurally. Okay, so this is telling me this protein function. Okay, this protein can bind structurally similar heat shock proteins and functions to facilitate proper protein folding and to reduce levels of non-functional protein aggregates. Okay, there are two functions of this protein that the paragraph introduces. The first is it can bind to the similar heat shock proteins to facilitate protein folding, and then the other function is to reduce the protein aggregation. Okay, perfect. Good to know the functions of this protein, right? There could be a question that asks us about this protein, so it's good to know its functions. Okay, let's keep reading. The larger HSP110 T17 deletions, okay, this is the deletion that we drew here, right? This is the deletion that we drew. Okay, so what about this deletion? This deletion causes what? Causes exon 9 to be omitted from the final sequence during pre-mRNA processing. Okay, that makes sense, right? Because this region where the deletion can happen, intron 8, and then after intron 8 is exon 9, right? If you delete a lot of stuff in intron 8, then some stuff from exon 9 can be deleted as well. Okay, so that makes sense. Okay, all right, let's keep reading. This generates a premature stop codon in exon 10. Okay, resulting in mutant protein. Okay, HSP110 delta E9. Okay, so in my brain, I'll just say, okay, mutant protein is the delta E9 because HSP110, that's the part that's the same, right? So delta E9 is the mutant protein. Okay, all right, people with a MSI, CRC have a HSP110 delta E9 transcripts in cancerous tissue only. Okay, all right, so that's the second paragraph. It has a lot of information, right? But it's really not too bad. It's basically just talking about central dogma, right? The transcription of the mRNA, right? And then eventually the translation of, and then eventually the protein, and then they introduce the function of this good protein, right? And then it also introduces that how that we can get this mutant protein, right? The mutant protein is really due to the deletion in this intron 8 region, and then this deletion causes series of problems, right? Like the exon 9 can be deleted as well. The exon 10 can have a premature stop codon, right? All these things, a series of consequences of this deletion. All right. Okay. Let's look at the table 1. Table 1 shows, what is table 1? Table 1 shows with respect to T17 deletion size, okay? So this is indicating me that this is kind of like the independent variable of this table, right? So the T17 deletion size, different T17 deletion size can give us what? The different amount. So here, the amount of HSP110 delta E9 mRNA expressed, okay? Okay, so this is my dependent variable, okay? The result I want to see from the table, like as I have different T17 deletion size, how does it affect my mutant mRNA expression? Okay, all right, so blah blah blah. Okay, this is just saying the same thing. So this whole paragraph, it's no fancy at all. We can read it, but it's basically just saying this. Okay, I would have just skipped to the table legend, but we can read it. Okay, so let's reread it. Table 1 shows with respect to T17 deletion size, the amount of HSP110 delta E9 mRNA expressed relative to the total amount of HSP110 mRNA expressed in MSI CRC primary tumor cells. The total amount of HSP110 mRNA expressed includes both wild-type HSP110, HSP110 wild type, WT is wild type, right? And HSP110 delta E9 mRNA, okay? All right, it's always a good habit to just finish reading the paragraph. I'm not encouraging you not to finish reading, okay? All right, so table 1 legend says, effect of T17 deletion size relative expression of this mutant mRNA in the tumor cell, okay? All right, so basically here, this shows us as the deletion increases, as I delete 3 base pairs or 4 or 5 or 6 or 7 or 8, up to 8 base pairs, and that would affect the concentration of this mutant mRNA, right? Okay, so let's see the takeaway of this table. So the takeaway of this table is like, as I go downward, I have more deletion. Okay, as I have more deletion, what do I have? That equals to what? That equals to more this mutant mRNA or less. Well, we all see that it goes from 0 to 100, right? So it's more. Okay, so more deletion kind of equals to more delta E9. Okay, delta E9 is the mutant one. Okay, all right, that's all I get from table. That's it. I can move on. Okay, and then table 2. Table 2 summarizes what? How this HSP110 delta E9, this mutant one, okay, this is talking about the protein, okay, not the gene, not the mRNA, okay, how this protein affects this wild type protein function in cells transfected with both of them, okay. All right, table 2, effects of HSP110 delta E9 on this HSP110 wild type function, okay? So I can see here that really quickly, this is talking about protein aggregation, this is talking about cellular localization, this is talking about apoptosis, okay? So what about those 3 functions, okay? So the wild type gives me about 64% aggregation, whereas the mutant one gives me way more aggregation, right? And then when I have those combined together in the cell, I also have a lot of the aggregation as if I'm just having this mutant one, okay? So I would say this mutant one, delta E9, the more of a delta E9 I have, the more aggregation I have, okay? These are the direct relationship I derived from table 2, okay? But from the first column, I mean. And then the second column is talking about cellular localization. Let's dig into that, okay? So when I have this wild type, and of course this means like none, right? I don't have any of the wild type or the mutant one, right? I have nothing. I have nothing at all, okay? This is this, and then but I think we need to more focus on a comparison of the wild type and then the mutant one, right? So let's see the cellular localization. When I have the wild type, I have more localization, and then when I have the mutant one, I'm only doing the localization in the cytosol, right? And then when I have both, and I also do have only the localization in cytosol only. So this is telling me that this mutant one is causing what? It's causing less localization. So I'm not gonna highlight it so that I have this mindset that I know, okay, the highlighted ones are the direct relationships, right? Whereas the one that I didn't highlight, they're inversely related, okay? All right, and then let's move on to the last column, which is the apoptosis, okay? What about apoptosis? So wild type apoptosis, I have a 25, and then when I have this mutant one, ND is not determined, I don't know, but I can see that when they are combined together, I have a more apoptosis. So I can roughly say that, okay, it's also a direct relationship, right? As I have more this mutant delta E9, I will have a more apoptosis. So I am gonna highlight this, okay? So I know that the more mutant delta E9 you have, the more aggregation and the more the apoptosis, and then the less cellular localization, as I didn't highlight this, okay? All right, so that's just my personal way of analyzing this table, okay? And then let's move on to figure 1, okay? Figure 1 shows the stability of human CRC cells expressing either HSP110 delta E9 or the HSP110 wild type to produce tumors after injection into mice, okay? So this graph, the dependent variable is the time, right? As the time goes by, as the days go by, that's the tumor volume is the dependent variable that we're measuring, right? As days go by, how are the tumor volumes changed? Does it increase or does it decrease, right? So as we can see that The mutant one is this solid dot one, right? So it's this line that I highlighted. Okay, maybe it's there's some overlapping, but anyways, the other line is the wild type. So what we can say as time goes by, right? As time goes by, the tumor volume of, I mean, which one? Okay, let's just say which one has a more increasement of the tumor volume, right? It's the wild type, right? This wild type has a more volume increasement. Okay, perfect. So that's all I get from figure 1. Okay, and then now I can move on to the questions. Okay, all right. So question 1 says, based on the data, can you guys see that? Based on the data presented in the passage, which statement best describes this HSP110 delta E9 allele? Okay, so this question is asking about which statement best subscribe about this mutant one, right? And then basically I glanced my answer choices, and it's asking me if it's either cancer promoting or cancer suppressing, right? And if it's dominant to this wild type or recessive to this wild type, right? So we can talk about the cancer part first, okay? So really the table 2 and figure 1 both tells us about if it's cancer promoting or cancer suppressing, okay? So as you can see here, this mutant one, delta E9, actually has a less tumor volume, right? As you can see from this graph, less tumor volume, also more what? As I highlight here, more apoptosis, right? Apoptosis means what? Apoptosis means like cell program death. So if cell are programmed to die, then you won't have this accumulation of bad cells, right? And then those accumulation of bad cells is cancer, is tumor. So both table 2 and figure 1 tells me that my delta E9 is cancer suppressing. Okay, and then is it dominant or recessive to the wild type? So as we can see that it's dominant to the wild type, because when I have this mutant one, I have 96% of aggregation. And then when I mix it up with the wild type one, I'm not getting like an average of 64 plus 96 and then divided by 2. I'm not getting an average number, but I'm just getting a number that's really close to what the mutant one would cause, right? So that means that this is dominant to the wild type one. Okay, so I'm gonna pick one that says suppressing and dominant. Okay, so C is my answer. Okay, and then question 2. Based on the passage, in MSI CRC primary tumor cells, the size of the deletion in the HSP110 T17 microsatellite is inversely correlated with. Okay, so our takeaway from the table 1, we said that more deletion will give us more delta E9 expression, right? So this is directly related, but the question is asking about inversely related to what, right? So I would say the opposite of this will be more wild type. Okay, so I expect to see this in my answer choices. Okay, that's even before I read any of the answer choices. Okay, so A says what? A says the amount of HSP110 delta E9 protein expressed, right? That's not inversely related. As we said, this is directly related. More deletion, more of this mutant one that's produced. Okay, that's expressed. Okay, and then B says the number of mature HSP110 wild type transcripts synthesized. Okay, so that meets my assumption, meets my logic before I digged into the answer choices. So I think this is really a good one that I want to pick. Okay, but you always want to finish reading all the answer choices. So let's just keep reading, okay? And make sure the other answer choices are not our best answers. Then we can pick B, okay? C says, the frequency of the omission of HSP110 exon 9 during splicing, okay? So the paragraph 2 talked about exon 9 omission, right? So this is really what? This is really happening when deletion happens. So this is also directly related to deletion, okay? So this is not my answer choice, my best answer. And then D says, the extent of premature translation termination in exon 10, okay? So this is also mentioned in paragraph 2 as we discussed the deletion, right? The deletion of 3 to 8 base pairs in blah, 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 in intron 8, that would cause the omission of exon 9 and as well as the stop codon in exon 10, right? So stop codon in exon 10 is just another way to say translation termination in exon 10. So that's also a direct relationship, right? So I decide that B is my best answer, okay? Because I struck through A and C and D, okay? All right, question 3. A man with a CRC mutation that results in the synthesis of HSP110 delta E9 and a woman that does not carry this mutation. All right, so this man, there's a law. So this man, this dad has this mutation. Then the mom does not carry this mutation. Okay, what is the % chance that the child will inherit the CRC mutation? So if you're not a super experienced MCAT test taker, you might struggle with the Punnett square now. You're going to say, okay, the father is probably AA and the mom is AA, right? And then you're going to do this. You're going to do this to figure out the, oh, there's a 50%, right? And then you really want to choose C, right? But how do you know that the father is a big A, small a? The father could be this. The question never tells me, right? I just know that the mom doesn't carry any of the mutation, but the dad can really be AA, right? And then you see this. Okay, so that gives what? 100%. So is it C or D, right? So some students may have this kind of struggle, so I'm showing you. But actually, you don't need to do this at all. Because why? Because as we read the first sentence that this is colon cancer, right? And then they're only I think, where does it say? Okay, here. This mutant transcripts only shows in cancerous tissue as well, right? So all these are telling us this is not the sex chromosomes. They are not on the X or Y sex chromosomes that can be inherited by the offspring. Whereas those are really the somatic, right? Those are, I don't know which chromosome is it on, but I know that it's not gonna be passed on to the offspring. So really, it's 0%. Okay, so no need to struggle with all this. Okay, okay, and then if you really did struggle with the Punnett square, you would notice that, okay, that doesn't give me either C or D, and then maybe I need to think in some other different direction, and then that might lead you to the thinking of, oh, this is not inherited by offspring. Okay, hopefully. All right, question 4. Which nucleotide pairings would be recognized by the MMR system during DNA replication? Okay, so at this point, my brain is so tired. I don't remember what is MMR. It's okay. I highlight that, right? It's the acronym. So, okay, MMR. Okay, in the first paragraph, defects in the mismatch repair system that corrects certain DNA replication errors. Okay, that's what MMR is, right? It corrects DNA replication. So, which is to say, if there was some replication error, then MMR will take action to repair it, right? So I expect my answer choices to be something that contains a mismatch, a DNA replication error, or more than one DNA replication error. I'll see. There are 3, right? 1, 2, 3. Okay, so the first one is matching T with C. This is what? This is wrong, right? This is wrong. This is a mismatch that MMR would recognize, of course, because the job of MMR is to recognize those mismatch, right? Because we know A, T, C, and G, right? So how I memorize this is like apple tree and then car in the garage, okay? And then how I memorize like A and T, they have 2 hydrogen bonds, and C and G have 3 hydrogen bonds. I remember that, okay, if I had a house, then I only want 2 apple trees, but I want 3 cars in my garage. Okay, I want more cars than apple trees. So yeah, more hydrogen bonds. Okay, so yeah, a little bit. Okay, let's go back to this question. Okay, number 2 is G and the A. Okay, that's also a what? A mismatch. Okay, what about the third one? A and T. There we go, our apple tree, right? So it's 1 and 2 that will be recognized by MMR, and then that is answer choice C. Okay, all right, question 5. The information in the passage best supports the conclusion that intron 8 of HSP110 most likely contains which of the following? Okay, so intron 8. I think we drew a picture of intron 8, all right? Intron 8, as the picture here we drew, intron 8 has a lot of this T stuff, this small repeated thymine that can be deleted, right? Deletion of 3 to 8 base pairs, okay? So this is my intron 8. And then what is the question asking about? Concludes that intron 8 of this most likely contains, oh, okay. So I know that intron 8 contains a bunch of T's, right? Which could be deleted. But what else does it contain? Let's read the answer choices. A says stop codon. Okay, I don't think stop codon will be in the intron, right? A is eliminated, okay? And B, splice acceptor site. I don't know about that. I just keep reading. And then HSP 110 gene promoter. Okay, that is wrong because we know that the promoter, if you have the background knowledge, promoter is always upstream of intron. Okay, so it's a promoter and then intron and then axon and then intron and then axon. Okay, so the promoter is wrong. Okay, and then D says partial coding sequence of this. This cannot be true because it's intron. Intron are the ones that are gotten rid of, right? The axons are the ones that exit the nucleus. Further, the translation can happen. So the coding sequence is obviously not in any of the intron, right? Because they're on the axon, okay? So D is wrong. So I don't really, even if I don't have the background knowledge about the splice acceptor site, I can do the eliminations and then say that, okay, B is my choice, right? B is, and then just so you know that, like I said, it's intron and then axon and then intron. So the alternative splicing or just the regular splicing, the acceptor site is about this region, okay? So that this spliceosome, which is the enzyme that cuts this mRNA, that can just get rid of my intron, okay? So this is where it cuts, and then this is the site called acceptor site, so that the exons can be made, okay? All right. Last one, question 6. According to the information in the passage, HSP 110 is most likely which type of protein, okay? So remember, we read about the function of the protein. If you still remember, there are 2 functions of that protein that we highlighted. One is, let me use a different color. One is what? Protein folding. Another is reduced protein aggregation, right? So I expect to pick an enzyme, pick a protein that will do this protein folding, or if I know any of the protein that can reduce the protein aggregation, okay? So A says adhesion, okay? That doesn't sound right, okay? Adhesion proteins, they're the ones that facilitate cell-cell adhesion, right? So I would like to eliminate this. And then chaperone, okay? And I have this background knowledge that I know chaperone facilitates protein folding. But just imagine, let's just imagine that you don't have this background knowledge. Let's keep reading, okay? So C says clathrin, okay? And I think you might remember from your cell biology or molecular biology class that clathrin is the vesicle coat that wraps up the molecule, and then this vesicle then can travel inside the cell, right? You might have heard of clathrin mediated endocytosis, okay? So I mean, this really is more about transportation instead of protein folding or any reduction of aggregation. So I can eliminate C, right? And then D is an enzyme. Well, I mean, enzymes are proteins, that's true, right? But this is really facilitating, this protein is really facilitating other protein folding, right? So I don't think it's a catalytic reaction by saying protein folding, okay? So I can eliminate D as well and then come to the conclusion that B is my best answer, okay? But if you have this background knowledge that you know chaperone is the protein folding protein, then you can quickly pick the answer, okay? So that's a taste of MCAT. I mean, we never done that during the 4 year of undergraduate science courses, right? It's more memorization, whereas MCAT really requires more critical analysis and reasonings. And I hope this is helpful.